We're going to keep our conversation about the physiology of nervous system going. And since we have discussed the small molecule neurotransmitters, I wanted to mention some of them. Neuropeptides, or large molecules. Opposite to small ones, these guys have long-lasting effects. Okay? And since they're so large, they act slowly. So you can imagine that these neurotransmitters are likely to regulate the responses that do not require an immediate response, an immediate stimulation, an immediate answer. Think about this. Chemical like acetylcholine, which is small and can be released in a very rapid fashion and stimulate, for instance, muscles very quickly, is very convenient in terms of the voluntary motor responses. You know, if I throw something at you, physically, I mean throw something at you, you want to be able to catch it in the matter of seconds. Okay? Now, when, say, you consume food, activation of smooth muscles in your stomach or your intestines doesn't have to be immediate. Okay? Your smooth muscles, everything's going to be just fine if you get them activated in the matter of, I don't know, minutes. Okay? But the effects of that stimulation usually require a longer time, so the effects are going to be prolonged. The first group of neurotransmitters that I want to mention is endorphins and enkephalins. They are inhibitory. They act in the parts of the brain such as hypothalamus and limbic system and brain stem. So parts of the brain that are responsible for subconscious control, subconscious, not voluntary activities. And endorphins are natural opiates. They inhibit the actions of substance P, and substance P, on the other hand, we kind of have to go hand in hand. Substance P belongs to tachykinins. The substance P is responsible for the transmission of pain. Okay? So it's excitatory. Endorphins that are inhibitory, they block substance P. So they inhibit pain. Does that make sense? Just that idea. So they work in opposite directions. Okay? Now, interesting thing is that endorphins are produced during physical exercise. Whether it's weightlifting or running or swimming. I don't know, if you do any type of training, you know how sometimes you perform exercises and it feels absolutely fine, and the next day you cannot move any of your limbs. Okay? I personally experienced this, you know, I busted my knee during one of the races really bad. I was just fine. And then it started to hurt. Okay? I saw people running, like losing their nails during the race, like nails completely just slawed off, and they just kept going, okay, during the ultramarathon. And of course, you know, next day you can barely walk if you can at all. So endorphins, they do inhibit pain. You may have, yeah. Shoes, because shoes suck. Yeah, fingernails. Yeah, fingernails. Like on the toes, the big toes. No. I mean, unless they run on their hands. No, it's the nails on the big toes. Yeah, I experienced that myself. Like after one of the races, the ultra races, they had podiatrists at the finish line, and they just looked at it and said, "Dude, we're just gonna cut them off. They're just hanging off. You lost them. Well, they grow back. Takes about 6 months." Yes. Yeah, there is. You should put that person that you want to protect in a tank with a cement, so this person cannot run, possibly. Really, I mean, I saw people running 100 mile, and at 50 mile they have that point when they take care of your feet, and it's just easy, in my opinion, just easy to cut the feet off. They horribly messed up, huh? Well, how are you supposed to run with what you have instead of your feet? It's horrible. You have another 50 miles in front of you. So yeah, but they don't feel that pain, and I can vouch for that. You know, when you run, you don't feel the pain that much, or you can tolerate it to some extent. And then when you take your shoes off and you look at this, holy crap, what's going on here? How am I supposed to walk? You cannot wear anything except for flip-flops for a week or something. Now, the problem with yeah, they are produced naturally. And that actually, you know, that term runner's high, that endorphins. Yeah. Well, there's a substitution for it called heroin. Okay? Heroin interacts with the receptors for endorphins. And it interacts much better and induces the signal that is much stronger than the natural signal coming from interaction between endorphin receptor and endorphin itself. What it ends up with is that natural production of endorphins, so endogenous opiates, natural opiates, their production ceases. When a person uses opiates like methadone or heroin or morphine or Vicodin for that matter, oxycodone, natural production goes down. And when that person decides to quit, the exogenous endorphins, exogenous opiates are not supplied anymore, and production of endogenous ones is gone. So there is no pain suppression, and let me tell you something, they do regulate the mood. I often use my experiences. I had 2 teeth removed once, 2 molars, and they told me if ibuprofen doesn't work, take oxycodone. And ibuprofen didn't work. And I took 1 pill. I can tell you absolutely sincerely, it was awesome. Really. I understand how people get hooked up. That's why I switched to ibuprofen immediately. It was so good. And it's not even heroin. So I understand why you want to do another one. And it's just a prescription drug, okay? So they do regulate the mood. I see how people get hooked up on that. Okay, and it's not just on the psychological level, and then add physiological parts to it, because you cannot quit. So simply don't. Yes.
She's just walking, talking, laughing, and then say that her pain is at 8. She's clearly becoming addicted. But just like you got prescribed something for pain, when you actually needed it, I feel like my tooth hurts a little bit, my belly hurts a little bit, and then it's just like that. That's reason to justify. Hey, I need this.
Yes.
So she got put back.
So it's now an epidemic. The Yes, well, the problem with heroin is that two major reasons. First, it's not clean. Second, it's injectable. So those are problems, Heroin was sold as the painkiller in the beginning of 20th century. It was used as the painkiller when we didn't know that it's addictive. Now, other neuropeptides, for instance, somatostatin, okay? It's inhibitory and it inhibits the release of growth hormone, pretty much regulates the process of body growth. Cholecystokinin is another great example. It actually has two completely different types of regulation. One regulates anxiety, okay? On the other hand, it inhibits the appetite. And it regulates the responses in the digestive system. It regulates the responses from the liver and the pancreas towards the food when food gets into the intestine. And obviously, if you, oh no, you didn't have it. I had a chance to look at the study guide because I wrote it. I can tell you, for the neurotransmitters that we discussed, you have to know whether they are inhibitory or excitatory or both, among other things. The ones that I don't mention here, obviously, you don't have to know. Does that make sense? You're not going to get a question, is neuropeptide Y inhibitory or is it excitatory? Here, I provided some glimpses into neurotransmitters that regulate the food intake. For some bizarre reason, a lot of people in our department are really curious about the causes of obesity and how the food intake is regulated and how can you prevent obesity. I have my own little recipe, how to prevent obesity. You have to eat less. But it's what I want to show you, it's not that simple, really, you know, because a lot of appetite regulation comes from the subconscious signaling in the parts of the brain like hypothalamus. Okay, it's physiological responses. For instance, there are diseases called chronic wasting disease and opposite of it, hypothalamic obesity, when certain parts of the brain are damaged and you have either overproduction of melanocortins or overproduction and oversignaling of agouti-related peptide or neuropeptide Y. Now, neuropeptide Y is an interesting chemical that increases the food intake. It is produced in certain areas of hypothalamus, so-called arcuate nucleus. It increases not only food intake, it also increases energy storage. How do we store energy, by the way? In the fat. Yeah, so it makes it fatter. At the same time, makes us less anxious. So it may partially explain why we eat when we are stressed. Does that make sense? Agouti-related peptide pretty much has the only function to increase the food intake. Now, two opposite chemicals, melanocortin and this horribly long-named protein called CART, okay? Cocaine and methamphetamine regulated transcript. You know, I always wonder about the brains of scientists when they were naming things. So the least creative is neuropeptide Y. Substance P. Really? Nothing better? Just substance P. And here, what happened? They detected, I bet what happened is they were studying the regulation of some physiological function in the brain, and they detected that this particular gene is upregulated when mice or rats or whatever were injected with cocaine or methamphetamine. And they said, that's transcript that's regulated by these two compounds. Now, obviously, this CART protein decreases feeding, which actually helps to explain how use of drugs, especially methamphetamines and CART and cocaine, can change feeding patterns. It can make people less hungry. Does that make sense? Decrease the food intake. So this kind of discussion of neurotransmitters kind of wraps up a conversation about synapses. And now we start to talk about hardcore physiology. That's the part that students probably like the least. I like it a lot because it's fun. We're going to talk about the mechanisms by which action potential, the signal in the neuron, is formed and transmitted. But first, we have to talk about the extracellular and intracellular fluid. So, we are walking colloidal solutions. We contain about 45 liters of water, each of us. 2/3 of the fluid that we have is inside of the cells, and 1/3 is in the outside. The fluid outside of the cell. Now, I want you to understand this terminology. Extracellular fluid is all fluid outside of the cells. Extracellular fluid can be found in two compartments. One compartment is plasma. Where is plasma? In the blood. It's a part of the blood. Another compartment is interstitial fluid. Where is interstitial fluid? In the tissues. Are we clear on that? Now, before we move on, if you change something in the plasma, what's going to happen to interstitial fluid? It's going to change as well. If there is any sort of change in interstitial fluid, what's going to happen with plasma? It's going to change. You see what I'm saying? It's not immediate. It takes a little bit of time. But plasma and interstitial fluid do communicate. Are we clear on that? Good? Now, I told you, or maybe I did not, and if I didn't, then I do it now, that this course contains very little math or numbers. I try to avoid number memorization. There will be some, but I try to limit it. This is one of the examples where I don't care about the numbers as much, but I do care about the ratio. What do I mean by that? I'm gonna walk you through this graph, and you will have to know the relationships between the extra and intracellular concentrations of certain ions. Does it make sense? Let's take a glance. On this graph, you can see red and blue bars that represent the concentration of ions outside of the cell. Red bar represents concentration in the plasma. Blue bar, concentration in the interstitial fluid. Yellow bars represent the ion concentration inside of the cell. Let me go back and ask you one little question about homeostasis. What is homeostasis? Period. Stop. Dynamic equilibrium. First, it's equilibrium, right? So it's sort of in a most stable state. But what I think is even more important, it is dynamic. So these bars, you have to appreciate, it's not that they like solid every second of your life. You see what I'm trying to say? Those numbers, those concentrations can fluctuate a little bit. Does that make sense? Are we good on that? So I want you to keep it in mind. Now, if you would look at this graph first and foremost, what you can tell about the concentrations of ions in the plasma and in the interstitial fluid. I mean, for all the ions. What? They're very close to one another. You see? They close, they close, they close, they close, and so on and so forth. Okay? The only difference is plasma. Okay, proteins, I'm sorry. Concentration of proteins in plasma is much higher than concentration of proteins in the interstitial fluid. Okay? Are we good? Is that understood? Awesome. Speaking of ions, know the ratios for concentrations of everything here except sulfate. There's a reason for it, believe me. Okay? Now, my suggestion, know it by exception. What I mean by that? There are 2 cations and 1 anion which concentrations are higher inside of the cell. Potassium, magnesium, and phosphate are inside of the cell. Everything else is outside. Does that make sense? Is that understood? So what? So say I'm asking you. So simple version of the question. Concentration of this ion is higher outside of the cell than inside. And I give you options: chloride, potassium, magnesium, phosphate. And you tell me it's outside more than inside. It's chloride. Concentration of the side is higher on the inside. And you tell me. More complicated question that I'm asking you. This ion will diffuse through its channels into the cell. So you have to think diffusion. In diffusion, ions go from high concentration to low concentration. So I need to figure out which ion from the least has higher concentration outside so it can get into the cell. Does that make sense? Sort of, I ask you to make an extra step. Am I clear? Good? Is that clear? Awesome. So, remember, potassium, magnesium, and phosphate are high inside of the cell. All other relevant ions, higher outside. You don't need to know about sulfate. Can we move on? Sulfate is higher inside, but you don't have to know about it. I'm not going to ask you about sulfate, because we don't really run into sulfate. I don't think we run into sulfate anywhere, really. Definitely not in this part of the course. Okay? Potassium, magnesium, and phosphate. Okay? 3 ions that have concentrations inside of the cell higher than the concentrations outside. I have it in a study guide, don't worry. So listed there. And I will post the first chapter of the study guide, first part of the study guide today, like after the class. Okay? Now let's talk about ion channels a little bit. It's sort of a recollection of what you already learned about. Okay? But it's, worth knowing. So, ion channels, transmembrane proteins that facilitate the transport of the passive transport of ions across the cell membrane. Does that make sense? When we say passive transport, what does that mean? You don't need ATP. So there is no need in using energy, which means ions will move from concentration that is that's really easy to imagine if you think about ions as little tiny balls that you release from the slide. They will run by themselves without any energy, only if they go from high to low. Does that make sense? So channels facilitate the diffusion. Okay? Are we good about that? There are four major types of the channels. We're going to talk mostly about three of them. So first type of the channel is leaky channel. Leaky channel is like a hole in the wall, okay? You can go there in any direction, nothing controls the movement really. You see? So if the concentration of certain ion becomes high on one end of the channel, this ion will start diffuse through the channel to the other side of the membrane. Is that clear? Do you understand what I'm trying to say? Good. Another type of the channel is voltage gated. We already ran into that type of channel when we were talking about synaptic transmission and calcium influx. When the membrane potential changes, channel opens and allows ions to get from area with high concentration to the area with low concentration. Is that good? Third type of the channel is mechanically gated. Okay? Any sort of mechanical stimulus can open the channel, and ions will start, moving wherever, from area with high to area with low. Now, mechanical channels, where do you think you're going to find them? On which cells? That do what? What function? The what? Mechanical stimulus. Where are you going to find the mechanical stimulus? Good answer. You can. Yes. Stretching. Where else? The skin. Everywhere you have any kind of mechanical stimulus, you're going to find channels that are open by mechanical stimulation. Your skin, your muscles, your joints. Does that make sense? Everywhere you have a mechanical stimulation. Digestive system is the good answer. When food gets into the, say, stomach and stretches it, there's going to be stimulation, mechanical stimulation. Merkel cells, sensory cells in the epidermis. They have mechanical ion channels. All kinds of sensory receptors. And finally, ligand-gated or chemically gated channels. Those are the ones that we talked about when we mentioned neurotransmitter interaction with ionotropic receptors. Remember? So, acetylcholine binds to the receptor on the channel, opens it, and now ions can pass through. Does that make sense? It's a great question. No, it's not one way. Channels are not one way. Channels are like doors. Does that make sense? I mean, I can get out, I can get in. Doesn't really matter. Okay? Now think about it. At the end of the class, you all go through that door into the hallway. So you go from the area with a lot of people to the area with no people. In the beginning of the class, you all get from the hallway into the class. Area with a lot of people to the area with no people. Same goes for ions. Ions will diffuse from the area with high concentration to the area with low concentration. Wherever these areas are. Does that make sense? Okay? Got it? Yes. Excellent. Change in the membrane potential. Change in the voltage on the membrane. Membrane is charged. We didn't talk about the mechanisms, how this charge is formed, and we will in pretty much just a few minutes. Okay? I'm gonna move on. Can I? Do you want me? No? Good? So this is really hardcore physiological concept, Nernst potential, that we need to understand in order to understand the mechanism for resting membrane potential formation. So it is named after a German scientist, Walter Nernst, and I promise when we're done with Nernst potential, I will tell you a couple of anecdotes about Nernst. I like these anecdotes a lot. Imagine you have a nerve fiber. Look at the picture A. Okay? You have a fiber. It's a cell, essentially, right? This is the membrane. This is the membrane. This is inside. This is the outside. Do you follow me? Now, for a second, imagine that the membrane of the nerve fiber has only potassium channels, nothing else. So only potassium ions can cross the membrane through the channels. Are we clear? Okay? It's a hypothetical situation. So potassium concentration is higher where? Inside. So potassium will diffuse out. Yes, out of the cell. And you can see it here. It is shown in those red arrows. Okay, potassium diffuses outside of the cell. Okay, unfortunately, this picture kind of shows you all steps together, so I have to highlight step by step. It diffuses outside. Potassium is charged how? Positively. So when positive ions leave the cell, what happens to the charge inside of the cell? It becomes negative. Very good. So you see this negative charge on the inside. Does that make sense? Now, what happens between the positive charges and negative charges? What do they do? Bingo. So positively charged potassium chemically diffuses outside. Does that make sense? But at the same time, a rising negative charge here will attract potassium electrically back into the cell. Does that make sense? So there will be a chemically driven diffusion to the outside and electrically driven movement inside. Before we go any further, do you understand the idea? Now I'm going to tell you a very important statement. At a certain point, the flow of potassium ions outside and the flow of potassium ions inside will balance each other. Does that make sense to you? Which means there is no net movement of potassium. And I want you to understand it very clearly. It doesn't mean that ions stand still. They constantly moving in this hypothetical situation. But concentrations do not change. Do you understand that? Okay? Now, this moment when the flow of potassium chemical flow of potassium to the outside and electrically induced flow of potassium inside, when they balance each other out, will happen at the certain distribution of charges across the membrane. That distribution of charges, okay, is called membrane potential. It's actually called Nernst potential for potassium. Okay? Does that make sense? Now this potential is going to be negative. And we'll talk about calculation of it. Does that make sense? Are we good? You understand that? Okay? With sodium, now imagine all the same situation, exactly the same. We have nerve fiber, which membrane is permeable only for sodium ions. The concentration of sodium is higher outside of the cell. So sodium will diffuse into the cell, making the outside of the cell charge-wise negative, but then it will become attracted back to the outside, and that will induce electrical flow, well, electrically induced flow of sodium back to the outside of the cell. Does that make sense? Think about it. It's the same situation with potassium, just flipped. You got it? So when flow of sodium due to diffusion and flow of sodium due to the electrical attraction balance each other out, that is Nernst potential for sodium. Does that make sense? Nernst potential for sodium in 61 millivolt. So, Nernst equation, shown here, allows you to calculate Nernst potential for the ion. Okay? This is the Nernst potential. Now we're gonna try and calculate some generic Nernst potential. Okay? Do you follow me? So say we're gonna calculate Nernst potential for, let's do for chloride. Okay? First of all, chloride, is it positively charged or negatively charged? Negative. When you calculate it for a negatively charged ion, the sign here should be positive. So 61, there's some like 3 pages of calculations how to get to that 61. Okay, 61. And then it is multiplied by the logarithm. Can anyone tell me what the logarithm is? What is the logarithm? What's the meaning of logarithm? Okay, what's the logarithm of 10? I know nobody likes math, but that much? Okay, try to figure it out for me. Logarithm of 10 is 1. Logarithm of 100 is 2. Logarithm shows you the power. You have to put 10 into to get the number under the logarithm. Does that make sense? Logarithm of 10 is 1. Logarithm of 100 is 2. Logarithm of 1,000. 3, 10,000, 4. Now, what's the logarithm of 1? 0. Logarithm of 0.1. Negative 1, 0.01, negative 2. Does that make sense? Okay. Now let's go back for a second and look at chloride. Is that chloride? So concentration, I just approximate. Concentration outside of the cell is something like 118. Concentration inside of the cell is, what do you say, 5? Probably 5. Okay. I'm just trying to think how, the best. You know what? We're going to do it this way. 120, and it's going to be 12. You will see why I chose this. So, I don't know why. Whatever. So, Nernst potential equals, for chlorine, 61 multiplied by the logarithm of the concentration inside, which is 12, right? And outside, which is, we decided it's going to be 120. Okay? What is this ratio? 12 to 120. 1/10, yes. What's the logarithm of 1/10? Yep. Does that make sense? Well, logarithm of 1/10 is negative 1. Negative 1 multiplied by 61 gives you negative 61. Okay? Does that make sense? That's how you calculate Nernst potential. Now, why it is so important? Because when it comes down to a cell doesn't have only potassium channels, or only sodium channels, or only chloride channels, or only calcium channels. It has all of them, okay? And knowing the concentrations of ions inside and outside of the cell, you can calculate the membrane potential for a given cell in a given conditions. Does that make sense? Okay. Now, I decided not to be terribly cruel. You don't have to know this horrible equation, but you do have to know this one. Okay, really know it. And that's one situation when I can ask you to calculate something. Yes, you can use your calculators for it. I can provide you with the concentrations of ions outside and inside, and you will tell me what the Nernst potential is. Am I clear? So be able to calculate the Nernst potential. I mentioned it on the study guide, so know how to calculate it. If you want, we can try and do it. Okay, I will find some real data, give it to you, and let's do it probably not today because I don't have it just at hand, but I'll make it for Tuesday, and we can just do it on the whiteboard or for the recording, okay, how to calculate it. Does that make sense? We'll do it. So, resting membrane potential. I want you to know the ionic flow that contributes to the formation of resting membrane potential. So, two ions that contribute to the resting membrane potential the most are sodium and potassium. So what happens? First, okay, you have potassium channels, leaky potassium channels that I want really to highlight, leaky channels that constantly seep potassium out of the cell. So if we talk only about potassium, what is the Nernst potential for potassium? Negative 94, so it's very negative, right? Now, sodium channels that are leaky, let sodium in. What's the Nernst potential for sodium? Yes. So if you would try to kind of combine them equally and, minus 94 plus 61, what you're going to get? Negative 33. Turns out there are more potassium channels than sodium channels. Does that make sense? So potassium contributes to the resting membrane potential much more than sodium. Do you understand that? Just because there are more channels. Are we clear? So that makes resting membrane potential of a cell negative 70. Just because there are more potassium channels. Does that make sense? If it doesn't, I like to hear it does not. Go ahead. It does not. Hmm? Let me break your question in two parts. So question number one is why we have so many potassium channels. Unfortunately, there is no why question in biology. It's not because I'm being a jerk. It really isn't. It's pretty much the same as to ask why we have two legs. Because selective pressure when we were evolving was in the way that two legs were beneficial and, kind of better, or our ancestors were evolving. It's better to have two legs than six. Okay? That's the way we function. Okay? Second question, potassium versus sodium in the blood. Well, remember, blood, plasma, is extracellular fluid. In extracellular fluid, potassium is low and sodium is high. But in the intracellular fluid, it's all opposite. So question that Jacques asked is, why do we constantly kind of drop potassium? And that's absolutely true. Potassium in the blood is very tightly regulated. If you eat something potassium rich, like 5 pounds of bananas, you immediately release potassium with urine. It immediately gets filtered out in your kidneys. The reason for it is exactly this. If levels of potassium in the extracellular fluid increase, all membrane potential stuff is immediately depressed. Okay? Nerve transmission is depressed. Do you know what, everybody understands what the capital punishment is? What is the most common way of capital punishment in the United States? Lethal injection. What's the main component? It's a potassium chloride. Very high dose of potassium chloride causes ventricular fibrillation of the heart and the heart stops. That's it. Simple as that. Okay? Because it inhibits the proper nerve signal transmission. That make sense? So, potassium has to be tightly regulated outside of the cell because it messes up everything. We have lower number of sodium channels. With sodium, it's slightly different. I'm kind of veering off but with sodium concentration in the extracellular fluid doesn't change much. Even if you will try to eat a spoonful of sodium chloride, which is the regular table salt, your sodium concentration will not change a lot. Why? Mostly because you will immediately become thirsty and will drink a lot of water. You will increase sodium concentration in the extracellular fluid, but compensate this increase by consuming water and increasing the volume of extracellular fluid. Does that make sense? If sodium becomes concentrated in the ECF, you immediately dilute it. Okay? Did I answer? Good. So, that balance between leakage of potassium ions out of the cell and leakage of sodium ions into the cell is what creates the resting membrane potential. But you have to appreciate that if they just keep going, the concentrations will change, and to maintain the proper concentrations of sodium and potassium across the membrane, there is a pump, okay? Sodium-potassium pump, bless you. Sodium-potassium pump that restores the concentration. It is constantly pumping potassium back into the cell and sodium out. Is that clear? Okay? So this is the mechanism of the resting membrane potential formation. Please understand the function for sodium and potassium channels and sodium-potassium pump. What do they do in this process? Am I clear? Am I? Good. Now, what if a little bit more of positively charged ions get into the cell? What's going to happen to the membrane potential? It will become more positive or less negative, whatever you want. So that change towards more positive or less negative level is called depolarization. Clear? Now, how can it happen? You either let positively charged ions in, so you let more sodium to get into, or you allow more potassium to leave the cell. Does that make sense? Cool. If membrane potential decreases, this is called hyperpolarization. Okay? That may happen if you prevent sodium from getting in, okay? Or you stimulate potassium to leave, or you let chloride to get into the cell. Am I clear? Regarding depolarization and hyperpolarization, I want you to understand what's going to happen if you, for instance, I tell you, if I open calcium channels on the membrane, what's going to happen to the membrane potential? Is it going to become more positive or less positive? So you have to think about two things. First, the concentration of calcium is higher on which side of the membrane? Outside. Which means if I open the channels, where does the calcium go? In the cell. What is the calcium charge? Positive. So if positive ions get into the cell, what happens to the membrane potential? It depolarizes. Cell depolarizes. Does that make sense? It becomes more positive. That sort of question. We good? No. All of them are. All of them have negative resting membrane potential. All cells in the human body have negative resting membrane potential. Some cells have resting membrane potential a little higher than others. So like neurons, negative 65, negative 70. Some cells in the eye, if I remember correctly, it's bipolar cells, have resting membrane potential negative 40. But it's still negative. Absolutely. Now, we're going to talk about graded potential. A lot of new terms. So graded potential is a local depolarization of the membrane. Okay? It's the graded potential. If you have some kind of a stimulus, shown here, It can be chemical stimulus, it can be mechanical stimulus, it can be electrical stimulus. Stimulus that opens the sodium channels, okay? Does that make sense so far? Opens the sodium channels. If you open sodium channels, where does sodium go? In the cell. Does that make sense? Sodium's outside. It goes into the cell. What happens to the membrane at the site of the stimulus? Is it depolarized or hyperpolarized? Depolarized. So depolarization means that the local membrane potential is changed. Do you understand? Now, this change in the membrane potential opens voltage-gated channels adjacent to it. When voltage-gated sodium channels open, what happens to the sodium? Step by step. Voltage gate, you're right, step by step. Voltage gated channels for sodium are open. Where does sodium go? It comes in, adjacent ones. Sodium goes in, depolarizes the membrane, opens the adjacent channels. Those arrows show a slightly different thing. Okay? Adjacent channels open, sodium goes in. Okay? Again, membrane depolarizes. Depolarization opens more voltage-gated channels, so depolarization spreads. Does that make sense? Stimulus depolarizes the membrane. Depolarization opens the adjacent channels for sodium, which makes membrane more depolarized on the sides, which opens more channels. Does that make sense? Which makes membrane more depolarized, which opens more channels, and so on and so forth. Does that make sense? I usually compare it to the stone that you throw into the pond. Waves. Waves start to spread away from the stone. Does that make sense? What happens to those waves eventually? They disappear. Now, you have to appreciate that according to the second law of thermodynamics, during every process, there's an increase in entropy. So some energy is lost. That's why eventually that depolarization wave goes away. Does that make sense? Okay? Do we understand that? Now, imagine this. If it's an axon, or it's a soma, it doesn't really matter. If it's a membrane of the neuron, if graded potential goes away before it gets to all the way down to the axon, you have no transmission, right? Now, interesting feature about graded potential is that if it reaches the certain level, the whole process of depolarization of adjacent fragments of the membrane, this process becomes regulated by positive feedback. It becomes self-sustained. Does that make sense? For this, graded potential have to go over a certain threshold in terms of change of voltage. This is the threshold. So if it goes beyond, then it becomes the action potential. Then it becomes self-maintained. It essentially becomes, it starts to go via positive feedback. I usually use the analogy of the punches. Okay? Think about this. Every punch carries certain energy with it. Does that make sense? Now imagine 3-year-old starts to punch you in the chest. It's probably going to hurt a little, but you're not going to get knocked out. And this 3-year-old does 1,000 punches. Okay? It transmits a lot of energy. I mean, it's gonna hurt for a while, but you're gonna be fine. Now, next time, it's gonna be Mike Tyson, who does exactly just 1 punch that carries same amount of energy as 1,000 punches from 3-year-old. Okay? And your sternum is broken and you're in the hospital. Okay? Above the threshold. Okay? So that is the effect. Does that make sense? Okay? So we're gonna talk about action potential next time. What I want you to understand now is, first of all, that graded potential is local, and if it doesn't cross the certain threshold, it will fade out. And it will not be transmitted, it will not spread it. But if it goes above the threshold, then it becomes an action potential, self-sustained signal. Is that clear? Awesome. Now,